So when we look at the types of immunity that we're going to get for the T cells and the B cells, with this adaptive immunity, that, adapting to the foreign particle and creating the cells, we say that we get cellular immunity and humoral. Note that it's not humor like being funny, because in the olden days they used to refer to the fluids of the body as humors. So that meant the blood, the fluids, the plasma, that sort of thing. In a cell-mediated immunity, the cellular immunity, that lymphocyte, remember the T's and our B's, they can directly attack and destroy the foreign cell or the diseased host cell, because some of the invaders are looking for a host cell, and they work inside the cell. So they'll actually move in, put the chemicals into it, and use that to kill that foreign cell. In humoral, this means we created antibodies. Antibodies travel in the bloodstream. They can get into extracellular fluid. So they're not going to directly destroy that foreign cell. Their goal, tag it. And the reason for that, if you remember the difference in the shape, okay? So antibodies, they simply go tag the foreign object. They're going to work outside the cell because they're going to be in the fluids of the body. And what this allows for, it allows for destroying a pathogen at different stages of its life cycle. Remember how I talked about, like, for example, a virus that can get into the cell and work in two ways? A lot of times when we're dealing with, for example, bacteria, they can destroy the bacteria as the bacteria is surviving in the body, or it can try to attack the working of the bacteria as the bacteria replicates, for example. So their goal is to be working against something specific, maybe a particular E. coli bacteria. Everybody's heard of that, right? Okay. So when we have the cell mediated, where we're going to attack a cell directly, we're going to use cytotoxic T cells. Cyte means cell, toxic means deadly, okay? So this would be cytotoxic T cells. Helper T cells can be used, because we're going to find that not all the T cells that get formed have to actually just go attack. Some of them are considered helper cells. They're very important in the immune system. So B cells, macrophages, dendritic cells, a lot of other cells of the immune system, what they're going to do, they're now going to become antigen-presenting cells. So those cells that were able to take in that foreign particle and ingest it, if it's too much for them to handle, this is when they're going to push a piece of it through their cell membrane, and that is now an antigen-presenting cell. And it will now go to the nearest lymph node and present that foreign particle. If your body has fought it before, that response is quick. If the body has not encountered this particle before, it takes longer, that's 7 to 10 days, before you start feeling better. Now, your textbook is going to talk about MHC 1s and 2s. These are our major histocompatibility complexes. I'm not going to get that detailed. If you are going to become an immunologist, yes, you're going to need to know these. What they are: surface proteins. It's like a little set of surface proteins. They're going to be used for recognizing foreign molecules in the body. The ones that have this complex 1, these are going to be on nucleated cells. 2 is when we have an antigen-presenting cell. One of those macrophages that has digested some of that foreign particle and has pushed it through the membrane. The way that we're going to work to kill the pathogen is cell-mediated. Do you guys remember perforins? What did perforins do? They perforated the cell membrane, meaning created pores, created holes in it. What about granzymes? Do you remember those? Those that would get put into the cell for the cell to burst. Okay. How about interferons? Alex?
It would stop the viral replication process.
Okay. Those work to keep viruses from replicating. Tumor necrosis factor. This would be when these cells encounter cancer cells. And they're going to want to keep that, because remember, cancer is when cell growth is out of control. Okay? And their goal, kill those cells that have out of growth control.
That's for any kind of cancer?
Yes. The body, cancer is actually a genetic disease. Now, whether that means, okay, well, my mom had this cancer, my grandmama had this cancer, whatever the case might be, yes, that would be hereditary, like breast cancers, okay? But cancer overall, whether it's one that has shown up in family or not, attacks the DNA. Okay? DNA, when the cancer becomes full-blown, the genes on the chromosomes are turned off now. We have genes on chromosomes that actually control the division of the cell. Cancer happens, that is turned off. Okay, so either way you look at it, cancer is a genetic disease because it's operating at the genes. Yes, some of it is hereditary, not all of it is. So, we're going to see that the ways to help destroy the pathogen under cell-mediated, acting on the cell directly, are to get that cell to burst, cytolysis, Phagocytosis, meaning digesting of the cell, meaning that macrophage is given help as it encounters the foreign particles to digest them. And then apoptosis, programmed cell death. That's what apoptosis is. In this response that's going to occur at the cellular level, we generate memory cells. So those T cells in the first response takes longer, but the second time you get exposed, it happens much faster and quicker, and you probably don't even know that you have even encountered that pathogen again. So in this cell-mediated response, we have a macrophage that encounters a pathogen. It will consume it because it's a macrophage, eating it, all right? As it digests it, it will take a piece and put it onto, or push it through, so to speak, the cell membrane. And it has now become an antigen-presenting cell, okay? Because it's presenting a piece of the foreign cell. Helper cells, if they have been formed or if they're ready, if they have the receptor, will recognize that, and in the process of going through fighting that response, we have to create a memory cell. So that memory cell will get created, just a few, not a lot. The rest of them will go through their processes. Now, cytokines are created in the immune response to help cells do their job. Now, they're going to be these particles, these proteins, these pieces that get formed, and they will help go to other cells to do their job. Now, I have a friend right now whose father has been diagnosed with multiple types of cancer. Liver cancer, bone cancer, it's pretty much metastasized in a lot of areas, okay? And one of the things that will happen with a person who's been diagnosed with cancer, for example, in the later stages, they begin to lose weight. And so that's what's happening right now. Like in just a week's time, you know, he's lost 10 pounds. Since the beginning of the year, diagnosis and all this sort of stuff, he's lost over 50 pounds. And so she and I were both, you know, like, why is he losing the weight so quick? The cancer is at the advanced stages. Okay, there's not a lot that can be done at this point. And so I was looking it up, and I was like, well, what is causing this? Where are those nutrients going? Because these cells are working so hard to try to fight the cancer cells, it takes a lot of energy for the cells to produce the cytokines. But because it's so advanced, pretty much fighting a losing battle at this point, it's using just about all of the energy from his body to make these cytokines. And so that's why he's losing his weight, because it's not going to his, you know, function for his muscles or other organs. And that's why people begin to waste away at the advanced stages of cancer, because they're not getting energy to their muscle cells or anything like that. Actually, the body is demanding so much energy the muscle cells are beginning to break down, and they're now using protein to make energy. And do you remember how I said we never want